So now let's consider the role of Smchd1 or Smchd1 in the maintenance of X chromosome inactivation. I haven't mentioned Smchd1 before. It's an SMC-containing, hinge domain-containing protein, and that's how it gets its name, Smchd1. But for simplicity, I'm going to call it Smchd1 because it's easier to say. So the background information is that we know Smchd1 is bound to the inactive X chromosome. We can find it co-localizing with the inactive X chromosome right out at embryonic day 13.5 in a mouse. So since initiation of X inactivation happened at E5.5, and we know that you're into the maintenance phase of X inactivation by E8 or E9.5 of gestation, then by the time you're out at E13.5, you're well and truly into just the maintenance of X inactivation. And so Smchd1's localization during this period suggests that it may have some role in this maintenance of X inactivation. We also know in embryos that are null for Smchd1, the females die at around embryonic day 10 or 11. Again, this is in the maintenance phase of X inactivation, not the very initiation or early stages of X inactivation. Again, suggesting they make it through those initial stages, but fail at the last stage. So we know that in these cases, the summary of what we know is that both imprinted and random X inactivation are abnormal. And this differs from what I told you about with DNMT1. And this is accompanied, the imprinted defect is an imprinted X inactivation defect is accompanied by a placental abnormality. So when you look in these embryos, you can see that exist expression is normal and that exist can recruit polycomb regressive complex 2 normally as well. But there is very little DNA methylation that ever ends up on the inactive X chromosome. But since DNA methylation doesn't appear to be involved in the placental form of, or the imprinted form of X inactivation, presumably Smchd1 is doing something else other than somehow directing DNA methylation. And finally, what ends up resulting is that you get a failure of the transcriptional silencing on the chromosome that should be the inactive X. So I'm going to go through the same sort of embryology, the same crosses, and looking at the expression of an X-linked transgene for Smchd1 as I did with DNMT1. In this case, though, it wasn't a LAC-Z transgene. Rather, it's a green fluorescent protein transgene, or a GFP transgene. So this GFP transgene is on the X chromosome, and it too is subject to X inactivation, like the LAC-Z transgene. And so we can use the expression of green fluorescent protein, which is of course very easy to detect under a fluorescent light, to indicate the activity of the X chromosome on which it resides. So there's an additional complicating factor in the genetics of these embryos. So on the paternal X chromosome, we have the GFP transgene, just like for DNMT1, there was the LAC-Z transgene. But now, in addition to that, on the maternal X chromosome, XM, we have an exist knockout. So since exist is the key determinant of X inactivation, if a chromosome doesn't possess exist because exist has been knocked out, then that chromosome cannot be inactivated. We know exist has to work in cis. So this maternal X chromosome, we know it has to be active. And this means that the paternal X chromosome, the one with the GFP, should be silent. So this is forced non-random, primary non-random X inactivation, and means that we expect that an embryo with this genetics up here will not be green and its placenta will not be green. So then we looked at these embryos that were either Smchd1 null or wild type. In the Smchd1 wild types, exactly as you expect, you find that the embryo is not green and the placenta is not green. However, when you look in the embryos that are Smchd1 null, you now find an entirely green embryo and an entirely green placenta. And this indicates that there's been a failure of X inactivation. And so X inactivation at some point has meant that you aren't able to silence this GFP on the paternal X chromosome. So this suggests that Smchd1 is required for the maintenance of both imprinted and random X inactivation. However, it's not formally proven because I haven't shown you that there was the detection of a previously active X chromosome, or previously inactive X chromosome. And I'll show you, this is instead shown in a slightly different set of experiments. So in this case, this reactivation of a previously silenced inactive X chromosome can be displayed in cells that are grown in culture rather than in embryos. And this has indeed been done not only for Smchd1 but also for DNMT1 and macroH2A, that histone variant that is enriched on the inactive X chromosome, among other genes as well. So here, fibroblasts are made, mouse embryonic fibroblasts, which are commonly called MEFs. These are made from mid-gestation embryos, E13.5 embryos. And remember, at embryonic day 13.5 in the mouse, you're well and truly just into this maintenance phase of X inactivation. X inactivation had been fully established several days earlier in development. So if these fibroblasts are grown from a mid-gestation embryo, and we have the same sort of genetics as we had in the previous slide, so we have already determined that the X-linked GFP transgene is on the inactive X chromosome, that is, that it should be always silenced. Now you can test whether or not you can promote the expression of GFP, so you can destabilize X inactivation. So what's found is if you have a control and you add in with that a drug called 5-azacitidine, this drug inhibits DNA methyltransferase 1, and so it inhibits this laying down of this final locking in inactive state. And if you do that, you really don't get any reactivation, even though you have destabilized some DNMT1. But if you do this in conjunction with a test, so if you do this in conjunction with either reducing the expression of Smchd1, reducing the expression of DNMT1, or reducing the expression of macroH2A, now in conjunction by having the drug and having the knockdown, reducing the expression through use of microRNAs, which I'd mentioned in previous lectures, then what you can do is you can reactivate GFP in a small proportion of the cells. It still doesn't happen very often because, as I said, maintenance of X inactivation is incredibly stable. But it does happen in a small proportion of cells because in combination, if you deplete DNA methyltransferase through a drug and either additionally deplete DNMT1 with a knockdown, with a microRNA against DNMT1 to reduce its expression, or remove the expression of some of Smchd1 or macroH2A, then in each case you can get upregulation or reactivation of that previously silenced X chromosome. And so this really serves to remind you that there are several layers that are locking in the inactive state on that inactive X chromosome once you reach the maintenance phase. And so really you need to destabilize them, and you need to destabilize not just Smchd1, but Smchd1 in addition to DNA methylation to be able to get this reactivation to occur. And that's because the reason that it's so stable, the reason it's actually hard to get it to occur at all, is because you have these redundant layers of epigenetic marks that are mitotically heritable. And this is really the key feature of the maintenance phase of X inactivation. In the next lecture, I'll go through a summary of X inactivation and all that I've told you so far this week.